Good afternoon. My name is Dr. Maria Nagy, and I'm the CEO of QuorumX Diagnostics. And I'm going to give you a presentation about our COVID Now technology, along with our pipeline that we have for QuorumX Diagnostics. And if you've already seen this presentation, just want you to know I will add the new details as we're going through the slides. So I hope that there's a lot of new information in there that you haven't heard. To begin, I'm talking about our COVID antigen test. It's a 4-minute test. That would be the fastest test on the market. The rest of them are anywhere between 15 and 30 minutes. Our COVID antigen test comes with a phone app that has videos embedded in there that will walk you through the test process, along with it will walk you through the test process, and in the end will read and tell you the results. Okay, as we all know, we're in the thick of the pandemic again, especially with this Delta variant. I read a report yesterday that said that the Delta variant, if you are unvaccinated and you get infected with the Delta variant, you're 2.6% more likely to end up in the hospital than with the original wild type. 2.6% more likely to end up in the hospital. So if you're not vaccinated, please, please, please go get vaccinated. You know? Okay, thank you. I know people that have yet to get vaccinated. Okay. All right. But with that said, even if you are vaccinated, you can get it and you can give it. So, yeah. But the vaccines will prevent you from going to the hospital. Okay, so we have developed a 4-minute at-home COVID antigen test. It's a lateral flow test, so it looks and acts just like a pregnancy test. We also have AI technology embedded in our phone app. So once you read the stick, once you get the stick and there's one line or two, you will be able to take a photo of that stick, and our AI technology will analyze the lines, and then it will indicate if you're positive or negative. Not only that, you will have a PDF report that you can share with either your doctor's office, your airport, if you're traveling, if you're going to a sporting event, if your employer is needing you to test, a school is needing you to test. There's a button right here. You can click and email that report away instantly. Lastly, we are working with Physician 360. So there is a button on the phone app that you can click immediately, regardless of your symptoms. It's optional. You can click this button right here, and it will connect you to the Physician 360 telemedicine service, and then they can give you more insight in terms of your symptoms and what you should do. Because as we've learned, everyone responds differently to COVID, right? Yeah. So our accuracy is 98.8%. The results are in 4 minutes. You do not need a prescription for this test. You can do this in the privacy and in the convenience of your home. You're not going to have to go to a location, have to wait in line, or risk infecting anyone else. You can do this at home. You don't have to send anything away. You can get results in 4 minutes, and then you can go on with your day. So originally when we did this testing, We were going to get approved for 18 and older. That was our goal. And then we were going to circle back around and test children. Well, one of the biggest feedbacks that FDA had for us is the fact we didn't test children. So they required or requested for us to open our testing back up. And so we've done that in the last couple of months. And now we can say, or we can probably say that our test is good for ages 3 and above. Our technology that we based this on was based on the technology that we had used with our other test, the QXD1 fast test for pseudomonal pneumonia. And so that's why in April, when we decided to focus on COVID, we were able to move so quickly because we had already learned the lessons and had a lot of experience working with these lateral flows. So we took what we had learned, applied it onto COVID, and we were able to move very, very quickly. I'm happy to say that our components are all custom. And when I say custom, when we developed our phone app, it is built from scratch, and we did our AI technology. We learned that when we did our AI technology, that when you take a picture of the lateral flow stick, you'll see this lateral flow stick. This one has an oval design. Well, when you get it directly from the factory, they come with a rectangle instead of an oval design. And they tend to be white in color. Well, our AI technology turns out it didn't like the rectangle, so it prefers the oval. So, and it didn't like the white, it preferred color. So we now, based on that information, and we were looking for oval cassettes or somebody that would be willing to sell us oval cassettes with color, and nobody was willing to do that. So we said, screw it, we're going to get our own custom mold made. So we're now proud owners of lateral flow cassette with oval, and we can add our own color. So we're quite excited about that. And we can utilize this not just for this technology, but for all our technologies moving down the pipeline. Our tube, we got that custom developed for us. We worked intimately with the plastics manufacturer for a long time, developing the most perfect tube. It took us 17 iterations. So the tube is specific for our product. You can drop a whole swab in there. It has a secure lid and a breakaway tab, and it's appropriately pliable so that you have a nice controlled drip at the bottom. And then not only did we work with our plastics manufacturer, we were also able to work with our swab manufacturer. And so after several discussions, we have a custom swab where we were able to get, it's pliable, yet the handle is sturdy. And then he was kind enough to make us a nodule at the 1-inch mark. So there's a little knob. So when you're putting the swab in your nose, that 1-inch mark is going to kind of guide the user so that they won't go too far in their nose. But they'll go far enough. So we're excited that all these pieces are custom. And not only does that help us with the uniqueness of our project, but it helps us with the supply chain because nobody else is going to be utilizing our components. So that will help us with supply chain. When we're discussing clinical trials, we distributed really over 450 kits. Now I think the number is up to 800. And we tested more than 350 people. It was done in an all-comer style, so we accepted everyone, regardless of age, regardless of comorbidities. We just accepted everyone, um, or regardless of their experience level with working with these at-home kits. These are all different kinds of questions that the FDA was asking about. But we accepted everyone. We tested everyone. And then we went to several different locations. People would call us up like, hey, there's a group of us at the gym. We think we got exposed, so we'd go test them, or they'd come to us. We went to the thrift store. We mailed a lot of kits for out of town and such. We had our own little drive-through where people would come and we'd bring out a little tray to them and they would test. Some people we tested through Zoom. And so we have a very nice representations of age and demographics and education and occupations. A very nice representation. We also utilize 3 different independent sources to help confirm our results. PCR, we had our PCR completed by Omni International in order to confirm our results. We worked with P23 laboratories for our FLEX and our LOD studies. And then we worked with Stat1 statistics to do our number crunching for us. So we try to utilize as many other sources than just ourselves to help confirm and to validate our results. When we're comparing the various technologies that are on the market right now, I think we've all heard there's Ellume, Quidel, Lucerna, Abbott, and then it'll be us. I think they're all over the counter, so we're over the counter as well. I think Lucerna's prescription. And everyone's antigen except for Lucerna. Interestingly, except for this molecular, molecular means that it's kind of like a PCR type of test, similar, but antigen means that they're looking for a protein. All these 3 companies, they're all looking for a nucleocapsid. We're the only ones that are looking for spike. And with that said, with the nucleocapsid, you have to break open the nucleocapsid to get to have access. You have to break open the virus to get access to the nucleocapsid protein inside. For us, you don't have to break open because we're looking for the spike, and the spike is on the exterior. So in our kits, in order to break it open, you're going to have to use some kind of toxic chemical, and luckily for us, we didn't have to do any of that kind of stuff. And again, utilizing some of the lessons that we learned in developing our other technology, we were able to get our test time, our processing time, to 4 minutes. And if you compare to the other people or to the other companies, we will be having the fastest test on the market. In terms of sensitivity and specificity, we're coming in at 96 for our sensitivity and 100% for our specificity. And then in terms of identifying the various variants, we are able to identify US, UK, South African. Lucerna is the only other company that puts those claims on their boxes. And we're really confident about the Delta variant as well, and we're in the process of confirming that in the laboratory as well. Okay, in terms of accuracy, like I said, our accuracy is 98.8 and our sensitivity is 96, 100% specificity. Okay. And when we're looking at the virus, here's the virus. And you see these yellow spikes right here. These are the spike proteins right here. And then over here, the protein, these purple ones are the M protein, and then you go in here And these these are the nucleocapsids. So they're on the interior. And like I said, you have to crack this open in order to get to the interior end protein, like, which is what these other companies are doing. And for us, we're sticking with the spike. So in the spike, if we were to lay that down and then if you lay it down and then you look at the RNA, there's all these different sections, and there's amino acids and or base pairs from 1 to 1355. There's two sections. There's S1 and the S2. S1 is the part that binds, it binds to the ACE2 receptor, right? So our antibody looks for this right here, right here. It will bind like Velcro to all these base pairs right here, okay? If we look a little bit closer, and we look at the alpha, beta, gamma, delta variants, and here we go, we're looking again at the strand over here, and then we look inside the S1 region, right here, receptor binding domain, RBD, right here is where the mutations are occurring. Now, mind you, our antibody recognizes all of this from 1 to 685 right here. And so if we're looking over here, the mutations, they're specifically in these like 5 base pairs, you know? And so the alpha one right here has this last base pair that has been mutated from the original. It's called the wild type, is the original. So the alpha has a variation right here in this last place. The beta has mutations here in the 1st, 4th, and 5th spot. The gamma is similar but different right here with a slightly different variation from beta in this 1st spot. And then here's the delta. And you'll see that there's 3 base pairs that have mutated. But we're pretty confident because, like I said, our antibody recognizes this whole area right here. And so we feel pretty confident that there's nothing to worry about when it comes to Delta. We, again, are confirming that in the laboratory. In terms of clinical trial, the big question is limit of detection. What is the minimum amount of virus in a sample that you can detect? Well, we did PCR studies and we did lateral flow studies with the live virus, or at least the lab that we worked with has done that for us. Here's 2 different patient samples, patient number 75 and patient number 14. This is positive and this is negative. And the interesting thing about this patient number 75 is that he came in, he's like, yes, I'm here to help you guys. I heard you guys were doing a study. I'm here to support y'all. But I did a test here a while ago when I was at the hospital last Friday, and I was negative. So, and I've been sitting at home this whole time for the last week. I haven't been out. So I know I'm negative, but you know, I want to support you guys. So we're like, thank you. Let's go ahead and run the test. He had no symptoms. Guess what? His stick came up positive, and we're like, oh my gosh. Because he's like, no, you know, I went a week ago and I was negative. And so we ran it again, and again, he came up positive. We're like, oh my gosh. And we're like, well, we are in the middle of a clinical trial, and it's going to take us a little while to confirm with our PCR. Go to CVS, go test, just to be on the safe side. So he went to CVS 3 days after he came to us. And guess what? He came up negative. He calls me, oh, I'm negative. I'm sorry, your test doesn't work. I'm like, oh, geez. You know, this is the first one that our results are inconsistent. Oh, darn. And Dr. Angel, he's like, no, don't worry about it. Maria, you know, have faith. Why are you so worried? I'm confident in our technology. I said, okay, let's see what the PCR says. He came back with the highest CT value of almost any patient that we saw in the entire study. So what had happened is he tested on this Friday. He came to us 7 days later, and then he tested again 3 days later at the CVS, and this was negative and this was negative. He must have gotten infected right here, and then he came to us and he was infected, and then he was negative, and we confirmed that with both PCRs when we tested, and they both came up positive. So, wow. When I say CT, he had the highest CT value, or let me rephrase, he had the lowest CT value. So CT, when you're doing PCR, what PCR is, you're taking a small fragment of RNA and you're amplifying it again and again and again and again, exponentially. And then at a certain point, you can use essentially a detector, and then you can measure this RNA. And if you only have to replicate a handful of times, then there was a lot of virus in there, a lot of viral particles. You only replicate a handful of times and you're already able to detect. But if you have a very small amount or no amount, you can replicate all the way up to 40 times and you're not going to see anything. So your measurements are going to be anywhere from 19 to 40, or maybe even 17. Guess what? He was 19. So, and then, so the question is, what is our limit of detection? So we caught Mr. 75 over here at about 19, right here. Here's 19 and here's 40. Once you get to 40, it's negative. So we caught here, and our weakest sample that we caught was 37.5, which is very close to 40. When you get anywhere from 36 to 40, you're crossing that line from the potential of it being positive to it being false positive, because you may have fragments in there that may come up as positive, but it would be a false positive. So there was a lot of talk, a lot of debate, or there has been a lot of debate between scientists. Should we make the cutoff 36? But we are able to, the lowest viral count that we could find, or that we've been able to detect, was 37.5. And then P23 in laboratories helped confirm that's 1.25 virons per microliter. So that's our limit of detection. So we were very excited about that. So that's our technology. Let's shift gears and let's talk about market potential, because you guys are investors and we need to see what is the potential market size. We've got 300 million Americans. Let's say only 10% are testing, okay? That's not the case right now, but let's just say only 10% of people are testing. And they're testing 2 to 3 times a week, like Dr. Fauci has recommended. So if you're doing like serial testing, that's a requirement of 60 to 90 million tests a week. And a single company is not able to produce that many tests, right? So the anticipated market demand is between 60 and 90 million tests. If we only cover 1 to 1.7%, that comes to 1 million tests a week. And that's where we max out based on the capacity that we have talked over with. Our capacity of our manufacturer will be 1 million tests a week. If we're selling that at $37.50, which is somewhere between our wholesale and our retail price, that comes out after a year of 1.95 billion with a 1.56 EBITDA. So the numbers are there. The numbers are high. Yeah. And the potential is high, and that's just only looking at the US. We haven't discussed the global need. And this is, yes, a global need. India has been calling me since November, and I can't do anything for them. Okay, over here, our go-to-market strategy, B2C, B2B, and B2B industry. So B2C will be direct to consumer on our website. Our B2B drugstore, we're going to do that. We'll be able to go nationwide within weeks and maybe even days of approval, since we have our 20,000 kits that are in process. Once we get that approval, Physician 360 will reach out to their 1,000 drugstores, and then we'll be able to get those kits out as quickly as possible to them. We didn't want to wait to order the kits until after approval, because if, let's say, approval comes September, then we have to wait another 2.5 months. We didn't want to do that, so we went ahead and ordered the kits. So that's how we'll get nationwide in drugstores. And then lastly, we have 4 salespeople ready to go. And they're going to focus on the government agencies, the schools, sporting venues, airlines. So they are ready. They're just waiting on approval. Here's our team. This is myself right here. Here's Dr. Angel Rivera, the guy that I keep talking about. Can't do this without him. Here's our very, very active advisors. They are always available, and again, we couldn't do it without their guidance and support. And then here's the rest of the team. We've got Jake McKinley, the app developer. We've got 3 of our 4 sales people right here. We've got Sherry, the director of marketing. We've got Juan and the rest of his team helping with the website and logo and the branding. And then we have our army of interns, and we definitely could not do it without the interns. And then just to let you know, we are not just a COVID company. We do have a very strong pipeline. Like I said, we've got this COVID now. We've got this pseudomonal pneumonia that we have the prototype ready. We were ready to go to clinicals with that, but we put this on hold. But that is ready to go. And then with our technology, we'll be able to do yeast infections, bacterial vaginosis, bronchitis, UTIs, whole myriad of other tests. And then right here, I'm going to squeeze in one more product, which is a COVID flu product. And after some discussion and some clever ideas, we think we can get to market very, very quickly with this COVID flu test. So, and it's going to require very little clinicals. So as soon as I can put some manpower on getting that put together, we'll get that ball rolling as well. Just to tell you about our patent situation, based on, I told you, our platform technology. Well, we just sent out our international utility patent application on that. We went through the PCT route, and once you apply to PCT, then you can apply to various countries. And so we did US. I guess that's not international, but it's US. We applied through that PCT application process, US, Canada, European Union, and India. So we have those patent applications in process, along with 2 provisional patents for our COVID product. I'm going to shift gears because I want to talk to you just a minute about our pseudomonal pneumonia technology. So on the pseudomonal pneumonia, this is looking for pneumonia that's caused by the organism Pseudomonas aeruginosa. Okay, and you see that in hospital and ventilator patients that have developed pneumonia. It's the #1, #2 cause. But more than that, this is a prototype, not just for that particular technology, but for our platform. And our platform is based on we're looking for unique proprietary biomarkers. These biomarkers, the name for them is called, well, they're the compounds that are used by bacteria to communicate to one another. And that communication process is called quorum sensing. So hence the name of the company is QuorumX Diagnostics. So when bacteria attack the body, they attack as an army. They don't attack as a single planktonic unit. They attack as an army. So in order to attack as an army, they have to communicate to one another. And so, you know, you got one guy saying, hey, are you guys all here? They're like, yes, we're all here. All right, so let's go ahead and attack. Okay, so first we're gonna launch our toxin #1, and then we're gonna launch our toxin #2. Hey, it's time to start developing a biofilm. All right, while you're developing the biofilm, well, let's make some antibiotics so that other bacteria can't attack us. And hey, let's make our efflux pumps. And so there's all this communication going on in not just one species, but in all these various species. And they each have their unique communication system. So if you can tap into that and you can listen to them, #1, you can identify which bacterial species you're talking about first. And then not only that, you can determine what their next course of action is. You can determine where they are in their bacterial infection cycle, which is big because when you dig into that, you may treat in the beginning of the infection differently than you do in the late stage of the infection. So if you can start understanding those different signals, which is what I focused all my studies on in grad school, if you understand those different signals and you understand the different languages, then you can develop different drugs and you can monitor infections in a completely different way than right now. We monitor infections by the body's response to the bacteria, not what the bacteria is planning on doing, right? So it allows us, you know, to develop these different lateral flow technologies. It helps in reduction of bacterial antibiotic resistance. And it's not just in bacteria, it's bacteria, viruses, fungi. And we also know that even in cancer. And so cancer cells use these different types of signals. As well. So Dr. Angel got very excited when he heard about that because his background's in cancer research. So when the time is right, I'm going to send him off with his team to go ahead and delve into that. And he's got a whole plan of various diagnostics that you can do around that technology once we get to, you know, get far enough past COVID to focus on these other things. And that it's for this platform technology that we got these international utility patent applications filed. So back in April is when originally we decided that we needed to start working on a COVID product. And so we opened a fundraiser on WeFunder to help fund this branch of the research. And so in April, we came to you with an idea and a concept, and we were able to raise between private investors and WeFunder investors $630,000. And so within that time, we were able to develop a prototype, get IRB approval, do an FDA pre-submission small batch run, build out a website, build a COVID phone app from scratch, do clinical trials, and now and apply to the FDA, and now reapplying to the FDA. So you entrusted us with this investment, and I think we delivered. And we've made some great progress. So we're asking you again to come in and trust us with some more investment. We have 4 goals this time. Number 1 is for 50,000. That will allow us to buy 20,000 units. Number 2 is for some COVID Now app upgrades. There's some things we built out on a basic build, and now we already know we want to do these more advanced builds. Goal number 3 is to build out the team. We need a CFO, we need customer service, we need accounting, we need dedicated science people to help develop these other technologies. And then lastly, it's 1.6 million, and that's so that we can buy the manufacturing equipment to increase our capacity from 1 million units a month to 4 million units a month. So those are our 4 goals. And I'm very excited to say that even though we don't open until Wednesday, we've already knocked out our first goal. We were able to order our 20,000 units. We already knocked out our second goal. We are already started on building the phone app upgrades. And so now we're working on goal number 3 so that we can build out the team and hire the interns, right? And lastly, I'd like to say, so we would hope, we need you to step in. We need you to invest and be a part of this, okay? And come and join the ride with us. And just remember, when you're investing in QXD, it's not just a COVID product, but it's platform technology, its pipeline, and its team. So thank you for your time. Good day. Reach out if you have any questions.